Welcome back to my YouTube channel. Today we're going to talk about the biochemistry of lipids. The way I want to structure this video is I want to talk about in these three main parts shown here on this image. That is, I want to talk about first the general properties of lipids at large. Second, I want to talk about synthesis of really just two different types of backbones that we can use to form other types of lipids. The biochemical synthesis, like the reactions, that sort of thing. And then thirdly, I want to tell you about a flow chart of the different categories of lipids and how they kind of relate to each other. But first, before I even start off with the general properties, which is the next page, I actually want to do one more thing, which is I want to orient any understanding that we have of lipids within the context of a bigger picture of biology or biochemistry at large. That is, I just want to introduce you to sort of bigger picture thinking, which is that whenever we think of biochemistry or just biology, we tend to think in terms of categories. That is, we tend to think of, for example, the organism being made of organ systems, and that being made of organs, that being made of tissue, that being made of cells, and so on and so forth. And so when we're talking about lipids, I just want you to understand lipids as being one of those categories of the things of which we are made of. Specifically, when we think of cells, we can think of cells as being made of organelles, and those organelles can be made of macromolecules, which are these really tiny macromolecules, and there's 4 categories of them, and lipids just so happens to be one of those categories by which we can enhance our understanding of biology at large. And just to mention them so that they're just included in the video or just for context, the other 4 macromolecules are proteins, nucleic acids, carbs, and lipids. Of course, nucleic acids has primarily to do with storage of DNA, which in turn translates over to proteins, and the proteins which can form numerous functions, most of which either being functional or structural. And then lastly, the 2 other ones are carbs and lipids. And both carbs and lipids can perform a variety of functions, both being either structural or functional. Though when we think of carbs and lipids, one way we can think of them is as being energy storage molecules, which they can be, and they primarily are thought of as being those things. And so when we think of carbs, we think of this fast-acting energy source, and then we think of lipids, we think of this long-term energy source. And we think of lipids, we also tend to think of it in terms of fats, and primarily the form of fat that we're going to store in our body is this molecule, which we're going to talk about in a second, called triglyceride, which is composed really primarily of two types of things rather, glycerol and fatty acids. And it's really the fatty acids that's part of this triglyceride molecule, which is also what we would think of when we say fat or solid fat, is what contains all of the energy that we are looking for when we consume fat and metabolize it. Okay, so let's talk about some of the general properties of lipids at large. So when we define lipids as being any sort of organic macromolecule which is insoluble in water. Meaning whenever you place it in water, it doesn't dissolve well, it's not very soluble in water. And this has to do with the fact that, this is a little background knowledge, but water is H2O, it has 1 oxygen, it has 2 hydrogens. The oxygen is covalently bonded to the hydrogens, however, due to the fact that the oxygen is much more electronegative than the hydrogens, meaning it has, if you want to personify it in a way, the oxygen has a greater desire to take more of the electrons that it's sharing with the hydrogens in its covalent bond. Therefore, that's exactly what ends up happening. In the covalent bond between oxygen and hydrogen, the oxygen takes more of, it's an unequal sharing of the electrons. The electrons tend to hang out more so in the oxygens as compared to the hydrogens in water. And because of this, you would call it polar. And one of the basic ideas of chemistry is that like dissolves like, meaning polar solvents only will dissolve in with polar solutes, and the same goes with non-polar things. So lipids are non-polar, therefore they are insoluble in water. And there's really 2 ways you can think about that too. You can either think about lipids as either being a lipid as being either hydrophobic or amphipathic. Hydrophobic just simply means that it is completely indissoluble in water all over. So every part of the molecule is, for the most part, doesn't dissolve well in water. Amphipathic, though, is actually something interesting. What it means is that there's a hydrophilic end and a hydrophobic end, meaning there's one end of the molecule of an individual singular unit molecule which is capable of dissolving in water well, due to its polar nature. However, the molecule is so large that there is another end of it which is hydrophobic, and it doesn't dissolve in water well. So basically you have this molecule which is, for the most part is insoluble in water, but some part of it is partially soluble. And so that's really what we mean by amphipathic. And that comes more into play primarily when you think of cellular membranes, because there's one part of it that, this bilayer of phospholipids, there is one part of it, namely the phosphate head group, that is polar and is dissolvable in water, and which sticks outwards towards the extracellular matrix and the intracellular matrix. And there is another part of it, which is the hydrocarbon fatty acid chain, which is hydrophobic, and it does not want to dissolve with water. That's a little too far ahead, but just know that a lipid is anything that is insoluble in water, and this can mean two things really. Either A, it is completely hydrophobic, every part of it is hydrophobic for the most part, or B, it is amphipathic, meaning there are some parts of it that is dissolvable in water and there are some parts of it that aren't. Second, this little neck part right here is basically a comparison between fat, protein, and carbs. Again, like I said earlier, there are 4 macromolecules. Essentially, we can really metabolize any of them to produce energy due to the fact that there is the presence of carbon-carbon bonds. And whenever we metabolize these for energy, essentially what we're really doing is we're oxidizing this macromolecule, this carbon organic molecule with these carbon bonds, and we're oxidizing it slowly but surely in a controlled and precise manner, such that the energy that is stored in these carbon-carbon bonds is being slowly but surely in a controlled manner released and converted into a more efficient form, while also increasing entropy. In the sense that there's a greater amount of entropy in the whole system. So because fat, proteins, and carbs all have carbon-carbon bonds, you can metabolize any of these to produce energy. And the idea of this little chart here is essentially to show that between the 3 of them, fat produces the most amount of energy per gram of what it is, per gram of fat. And the reason of this is because compare carbs and fat. Carbs, what is it? It's sugar. It has, its chemical structure has carbon, hydrogen, oxygens. The primary reason why fats can yield more energy per gram than a sugar is because it is in a more reduced state, meaning it is not already partially oxidized due to the fact that it has oxygen. Whenever we take the carb, we're going to oxidize it further so that we can release more of the energy and convert it into more usable and useful form. But the only problem with the carbs is that, in terms of the amount of energy per gram, is that it already has some oxygen in it. And it's because of this already presence of some oxygen that some amount of it is already oxidized. Whereas with fats, you don't actually, and again, when I say fat, I'm really referring to triglycerides. And when I'm saying triglycerides, I'm really referring to the fatty acid portion of the triglyceride, which we'll talk about in a second. This hydrocarbon chain, or the fatty acid, or the fat, it doesn't have any oxygens on it. Therefore, it's not already partially oxidized. Therefore, it is more so reduced. Therefore, there's more energy to be had that can be released. Another thing also, there is a bit of a trade-off. So fats can package much more tightly together. So you can, just like packaging wise, like if you're playing Tetris, it packages much nicely more together as compared to carbs, which is another good feature of it. However, the downside to the fat being constructed this way is that relative to carbs, you can burn and consume and metabolize the carbs much more faster in a much more efficient way as compared to fat. And the other downside is that carbs, because they are polar, they are hydrophilic, meaning they are soluble in water. Therefore, you can put it in the bloodstream, which is mostly water, and it will dissolve and it will be all right. However, with fat, the only problem with fat is that because it is, like I said, insoluble in water, and this can mean one of two things, amphipathic or hydrophobic, because of this fact, it doesn't dissolve in the water of the bloodstream very well. Therefore, that's why you have to have, and I'll talk about it in a minute, special molecules, these complex structured molecules composed of a lipid portion and a protein portion called lipoproteins, which carry fats in the bloodstream from one place to the other. So this part right here has to do with some of the functions. Of course, there's more than 5 that I've written down right here, but there's definitely more. I've just written down these here for simplicity and for trying to be as concise as possible. Firstly, like I said before, like I referenced before, you're made of an organism made of organ systems made of organs, and then from there you have tissues, cells, and cells are made of membranes. Practically everything in an organism is enclosed by some sort of membrane of some sort, and there's a very good reason for that. But when we think about the cellular membrane, they are composed of these molecules, this bilayer of phospholipids with a phosphate head group and a glycerol backbone and two fatty acid chains, which can be saturated or unsaturated depending on how fluid you want the membrane to be at different parts of it. But the idea that I want to point out here is that lipids are very, very important in the sense that they compose a very crucial and basic structure that every single cell is made of, which is the membrane. And it's due to its chemical makeup, the fact that it's amphipathic, and that just the way that it's made, that it allows it to do its job as a cellular membrane. Secondly, I want to talk about fat-soluble vitamins. So fat-soluble vitamins, due to the fact that unlike water-soluble vitamins, which you can consume and use much more fast, fat-soluble vitamins, the reason why they're called fat-soluble vitamins is because they cannot, like lipids, they're insoluble in water, therefore they're insoluble in the bloodstream, therefore they have to be carried by special lipoproteins to be carried to its target peripheral tissue or to be stored in either fat or the liver. And so one of the important functions, and this is the second function, of lipids is to act as lipoproteins to carry other macromolecules through the bloodstream, which otherwise they wouldn't be able to. So the third, fourth, and fifth points. So essentially we're going to talk about the fourth point, which is that lipids are a good macromolecular storage unit for energy. The third and fifth point have to do with the production of hormones. And the third one has to do with cholesterol, which is a hormone that can be further biosynthesized into other various steroids depending on what kind of organ is being done in. And fifthly, you can also use lipids to produce other non-cholesterol derived hormones called eicosanoids, which generally the eicosanoids they're derived from arachidonic acid, a 20 carbon polyunsaturated acid with fatty acid with 4 cis bonds, double bonds, from which you can develop these different eicosanoids, and the eicosanoids really just have to do with inflammation and that sort of thing. That's a little ahead of where we want to be right now though. So this little box right here has more so to do with the relationship between the structure of a fatty acid and its the strength of its intermolecular forces, and which in turn, the greater the intermolecular forces of a given fatty acid, which is based off of the structure, the greater its melting point. And the greater its melting point will determine whether it is a solid or a liquid at room temperature. If it's a higher melting point, that means it's more likely to be a solid. If it's a lower one, that means it's more likely to be a liquid. And again, weaker intermolecular forces, which again is just the strength of the forces between other triglycerides with each other, the more likely it is to have a lower melting point, therefore the more likely it is to be a liquid at room temperature. And so again, when we think of the 4 different types of intermolecular forces, there's really just 4 of them in order of increasing strength. They are van der Waals forces, also sometimes called London dispersion forces. Secondly, we have dipole-dipole forces. Thirdly, we have hydrogen bonding, and then fourthly, we have ion-dipole. I'm not going to talk about the others. The point I want to make is that because lipids, because specifically the fatty acids are non-polar, the only intermolecular force that they can generate is van der Waals forces. And also another thing I want to point out, or I'll say it in a second. So the first one, you see that the greater the molecular weight, in other words, the longer the hydrocarbon chain is, the greater the intermolecular forces and the greater the melting point. The reason for this is as the hydrocarbon chain of the fatty acid gets longer, there is more of it to be attracted to each other. And that's kind of the idea of van der Waals forces. The van der Waals forces is this idea that every molecule exerts it, and it's the idea that even for like a split second, electrons are basically moving around in these clouds of probability. For a split second, more of the electrons might be on one end as opposed to the other, and this produces a partial, momentary, temporary charge. And this charge can also influences other molecules around it to kind of do the same. And then when they have all these different charges, they will be more attracted to each other. It's not very strong. It's the weakest of the intermolecular forces, but the idea is that the bigger it is, or the longer the hydrocarbon fatty acid chain is, which is also another way of saying the molecular weight, the greater the intermolecular forces, the greater it is attracted to each other, which in turn increases its melting point. And then secondly, the second little row here is to say that the greater the unsaturation, or in other words, fatty acid chains, they're carbons bonded to other carbons, which also the carbons are also bonded to hydrogens. However, sometimes they can be unsaturated. And by unsaturated, I mean unsaturated with hydrogen. So they don't have as many hydrogens as they could have, meaning in place of that, in order to still remain neutral and to have 4 bonds total, it will substitute the lack of hydrogen, or the unsaturation of hydrogen, with a double bond between the carbons. And so essentially, unsaturation basically just means, in this context, that there is a double bond. And so what this relationship says is that the more double bonds there are, which also in turn means the less hydrogens there are in its place, this means that the intermolecular forces are weaker, therefore the melting point is weaker, therefore it's more likely to be a liquid at room temperature because its melting point is low. And the reason for this is because the double bonds produce kinks in the chain. In other words, it's no longer linear and straight, and for the most part, just linear and straight. When you have double bonds in it, and the more you have, the more kinks you introduce into the chain, and therefore it's just much less organized, and it can't exert the same amount of strength of van der Waals forces when it's as unorganized as it is, and it's no longer in proximity to each other, and they can't stack on each other as efficiently as they could otherwise. And so these last two little boxes here have to do with the ways that we will kind of categorize fatty acids. On the left here, you can see that fatty acids can be categorized based off of their lengths. If it has less than 6 carbons, it is short. If it has between 8 and 14, it is medium. And if it has 16 and 24, it is long. Most fatty acids are going to tend to be longer as opposed to shorter, though. And also, most fatty acids if they are unsaturated, meaning they do have the double bonds present along the fatty acid, they tend to be a specific isomer called cis as opposed to trans. Meaning that while it is true that for a double bond to occur along a fatty acid hydrocarbon chain, what this essentially means is that instead of each carbon having 2 hydrogens bonded to it, specifically for these 2 carbons that are double bonded to each other, instead of having 2 hydrogens each, they only have 1 hydrogen each in order to make up for the double bond between them. And what this means is that the hydrogens that each one of these carbons of the double bond, the hydrogens can either be cis or trans, meaning they can either be relative to each other, they can be on opposite sides or on the same side, like adjacent sides. Cis means that they're on the same side, stereochemically, that is. And then trans just means that they're on opposite sides. Naturally, natural fatty acids are cis. You only really get trans fats whenever you react the fatty acid, the unsaturated fatty acid, with hydrogen gas in the presence of a catalyst such as nickel. And this causes some amount of hydrogenation to either some or all of the double bonds, if it's polyunsaturated, if it's mono or unsaturated, whatever that might be. And the trans fats basically occurs in this process where you're trying to hydrogenate the double bonds of the fatty acid whenever you don't fully hydrogenate it. In other words, if there's impartial hydrogenation, that means that instead of going from a cis double bond to a single bond with more hydrogens, instead what you might end up is with a trans double bond. So you just converted it from one isomer to the other. And then lastly, this right here is just a way of writing. So different fatty acids, they have different names, like common names. This is just a way to show how you write it. The way that we write it, for example, in this first example, is this 18 and then 1, 9. 18, the first number, has to do with the number of carbons. The 1, or the number after it, if it is unsaturated, the number after this first number, which again the first number is the number of carbons, that number after it has to indicate the number of unsaturation. So the number of double bonds, in other words. In this case, you have 1, and then the last number that follows after is to show where along the carbon, the fatty acid, that this unsaturation is located. relative to where you start counting from the carboxylic end. And the first carbon would be the carboxylic carbon. And so here for the second example, for arachidonic acid, you have 20, which indicates the number of carbons, and then 4 is the number after this first number, which is the number of unsaturation, or the number of double bonds. And then this 5, 8, 11, 14 are all just like locations of, you know, carbon number 5, there's a double bond. Carbon number 8, there's a double bond, and so on and so forth. So that's just like a way of writing it. This version on the bottom here that says 24 and then delta 5, 8, 11, 14. Delta, that little triangle there, is just another way of writing the same thing, really. Okay, so this page and the next page are going to be about the specifics of the biochemical synthesis of different types of lipids. The way we're going to go about this, though, is we're going to talk about this in really in terms of two different backbones. In other words, there's going to be these two base molecules from which we're going to add things to get different types of lipids. And these two different types of backbones are the glycerol and a sphingosine. And so first we're going to talk about the glycerol, and then we'll talk about the sphingosine on the next page. Essentially, the idea of this is we're essentially going to take all of these different like Legos, these different like building blocks, and we're going to add them to each other via chemical reactions to create increasingly larger, increasingly complex lipids. And again, the sort of these are like 4 of the building blocks we're going to use. We're also going to use some other little building blocks, but these are just the 4 well, I'm going to name here. This is a phosphate group. This is one of the little building blocks we're going to use. This is a fatty acid. This is the hydrocarbon chain of it. And then this is the carboxylic acid portion of it. And this is the glycerol. I already said that part, and that's the sphingosine. So the way we're going to go about this, though, is we're going to, and by the way, what I mean by the way we're going to go about it, is the way that we're going to add them to each other has to do with this one big main idea mechanism, which is that we're going to take any kind of molecule with a carboxylic acid portion, or we're going to take a carboxylic acid portion, which is this, and we're going to react it with an alcohol portion, which is this, and they're going to add to each other such that they add to each other at these 2 points. And so really we can do this with 2 different, other than the fact that we're going to have an alcohol component right here, which is the blue one. This other component can be really either one of 2 things. It can either be this carboxylic acid, or it can be the phosphate group. And the basic idea is that this is a condensation reaction, by the way, meaning that we have 2 things that are adding to each other, and via the addition of each other, you're going to have the loss of water, which you can see on the far right here. The way that we're going to go about this for kind of both of them really is the first step involves the deprotonation of the hydroxyl group here, or the alcohol group here, such that there will be a negatively charged oxygen because you deprotonated and you removed the hydrogen. And then from there, the oxygen by definition, because of that, will be the nucleophile, and this carbonyl carbon right here at the carboxylic acid, this little inflection point right here, is going to, in this case, be the electrophile. In other words, this is the electron poor atom, this is the electron rich atom. And what's going to happen is that they're going to react and they're going to add to each other such that the oxygen is going to add to this carbonyl carbon right here. Before, you had a trigonal planar sort of shape right here for this carboxylic acid, but by adding the negatively charged oxygen to the electron poor, or in other words, electrophilic carbon here, you're going to form a tetrahedral intermediate structure. And then after adding the oxygen, and with the oxygen, any kind of R group, which is literally just anything that's added to the alcohol component, it could be anything. Afterwards, again, you form a tetrahedral intermediate, and by doing so, you break the double bond of the oxygen of the carboxylic acid so that you can form the tetrahedral intermediate without that center carbon being charged in any way. And then afterwards, after you form the tetrahedral intermediate, what's essentially going to happen is that this OH group, this alcohol part right here, is just going to leave. It's going to dissociate and leave. And you're going to reform the double bond. So essentially, you're going to deprotonate the alcohol part, and then it's going to add to the carboxylic acid, and then it's going to shift around and it's going to remove one of the original alcohol parts of the carboxylic acid. And then by doing so, you create something like this, where anything to the left of it and anything to the right of it could be any sort of R group, which we will see in soon is going to be specific R groups. And yeah, one way you can imagine this, again, this is just the basic idea of Fischer esterification. One way you can imagine this is they're going to add to each other these 2 points here, such as that this is one part and that's the other part. You can, if you wanted to imagine it, you can just draw the product by removing this and this hydrogen over here. So you just like draw a box and you like, whoop, and then this is the product. Whatever that wasn't in the box, they get added to each other. And so the same sort of idea applies to this second mechanism, which really is just a repeat of the first one, except this time instead of using carboxylic acid, you're using phosphate group. And in this case, it's just the same thing. The only difference is that it's a phosphate group, and instead of forming a tetrahedral intermediate, because the phosphate's already a tetrahedral, it would just form a trigonal bipyramidal intermediate, which then collapses as it removes one of the original alcohol hydroxyl groups as a leaving group, and then you get them two added to each other so that you create a phosphodiester in its place with the loss of water, or in other words, just H2O. And so essentially, this same concept is going to apply with, we're going to talk about this first in terms of the glycerol, and then we'll talk about it in terms of sphingosine. So this same sort of concept applies with the glycerol. Essentially, all we're going to do is we look at the glycerol, all it is just a carbon here, a carbon here, a carbon here, and to each carbon, you're also going to have a hydroxyl group. Essentially, what's going to happen is that you have these 3 points, these 2 hydroxyl groups, and all 3 of these are essentially just these opportunities for a carboxylic acid of the fatty acid to add to it. So that's all that really is. And what you can think about this in a few different ways, in the sense that you can add to it in different ways. So for example, here we have a triglyceride. This is what I was talking about earlier. This is the main storage of fat. This is the most sort of basic idea of a lipid in the most broad sense possible. Here, what you have here is the same reaction mechanism. In this case, the carboxylic acid in this case would be the fatty acid part, and then this alcohol part is going to be either any one of these hydroxyls of the glycerol. So essentially, the idea is just that in this case, if we only added one fatty acid to one of the hydroxyl groups of the glycerol, you would get a monoglyceride. If you added 2, you get a diglyceride, and then if you added 3, such that that's its max carrying capacity, you would get triglyceride. It's a very basic idea. It's the same mechanism for each one of these. It's all condensation reactions that involve deprotonation, addition to, and then followed by addition to the carbonyl carbon of the carboxylic acid, and then the expulsion of a leaving group, which is the original hydroxy group of that carboxylic acid. And so one interesting idea though is that we can actually do this in a couple different ways. And another way you can do this is, like I said, you have 3 hydroxy groups on this glycerol backbone. But what if you only use 2 of them for the fatty acid, and then the third one you used for a phosphate group? And that's kind of just what happens with this phospholipid here. All it is that you've got a phosphate head group, and you've got these two of the hydroxy groups of the glycerol being dedicated towards having a fatty acid reacted with it in a Fischer esterification-like manner. And so again, we can take this idea a bit of like a step further, in the sense of, instead of using a phosphate head group right here that is attached to one of the alcohols of the glycerol backbone, we can actually, in its place, instead of using the phosphate group, we can use a carbohydrate, like a monosaccharide, or we can use multiple, so polysaccharides. Let's talk about this in terms of monosaccharide for a second though. Essentially, what's going to happen is, again, it's the exact same idea. It's the exact same mechanism as right here, this first one right here. Or not exactly, it's mostly the same. So what we're going to do is we need to essentially render the carbohydrate to be an electrophile. And the way we do that is that you react a leaving group, which is this Lg right here, with the carbohydrate, and it basically attaches to the anomeric carbon's oxygen, or the oxygen of the anomeric the oxygen of the alcohol of the anomeric carbon, which is this one right here. And essentially what happens is that the leaving group causes the oxygen bond into the leaving group, which is this right here, to leave, and in its place you're going to form a double bond in its place and a positively charged oxygen atom because it has 3 connections as opposed to 2. And by doing this, by adding a leaving group and causing the leaving group to leave and create this double bond in a positively charged oxygen, essentially what you're doing with that is you're creating an electrophile in its place. In other words, it's an electron poor atom, and then the alcohol part of the glycerol, which is in this case would be right here, is now therefore the nucleophile, and essentially the electrophile and nucleophile react to each other, and basically they add to each other. That's all it really is. So imagine in this picture, instead of a phosphate group here, you have this carbohydrate. And again, the way we did that is we just reacted the carbohydrate with a leaving group to cause it to become a electrophile in its place. And that's really the idea. There's really only 2 backbones that we're going to be thinking about, at least so far as this video is concerned. That is the glycerol backbone, which we've already talked about, and the sphingosine backbone, which is what this page is about. Again, the same idea still remains. We're still going to use the same sort of reaction mechanisms or the same 2 reaction mechanisms which we talked about before, which are really just kind of the same one, just with different sort of either phosphate or a carboxylic acid. It's the same idea. We're just taking these different little building blocks, like these little Legos, if you want to think about it like that, and we're just adding them to each other to create these larger, more complicated molecules that will have different sort of chemical behavior in different environments. And just again, in this case, we're just using a sphingosine. Sphingosine, like I said before, it's this right here, or also if you wanted to look at this image, it's here. These are 2 of the same things. The way I think of sphingosine is essentially as just a glycerol with one of the middle hydroxyl group being a nitrogen instead, and as having a fatty acid with a singular double bond attaching to the fatty acid. Maybe that's a silly way of thinking about it, but that's just the way I think about it. So first, again, this right here is kind of just like a little flow chart showing the progression of different fatty acids, or not different fatty acids, different lipids that we could create by adding different components to it, and so on and so forth. The first one has to do with adding a fatty acid. Again, it's the same reaction mechanism, except instead of this being a hydroxyl group, the nitrogen is now the nucleophile, and is that whenever you react the fatty acid with the sphingosine, the fatty acid is going to add itself to this nitrogen right here. And then the idea here is that you can actually also add a phosphate. Like I said before, it's the same sort of idea. The phosphate can add to either one of these hydroxy groups. In this case, we have it to the top one. And then from there, like I said before, kind of like what we did before right here, where you can add an alcohol component to the phosphate head group just to make it bigger and more complicated. The same can be said here. I didn't draw it out because I was lazy, but just imagine that you could add any sort of alcohol with any sort of R group being whatever it is that you want it to be, and you can add it to the phosphate head group right here, such that you just elongate and create a bigger, larger, more complicated molecule. And then another possible sort of way you can go about this is you can actually add either a monosaccharide or polysaccharide to your sphingosine, which already has this fatty acid. And by doing so, you're really going to create one of 2 things. If you add only one singular sugar, in other words, just a monosaccharide, you're going to get a cerebroside. And if you add multiple, or in other words, polysaccharides, you're going to get a ganglioside. And the way I kind of remember that is the ganglioside, the first word you hear is gang, and gang is multiple people, therefore multiple sugars renders multiple people, I guess. And so that's really just how I think about it. Again, it's the exact same mechanisms that we're talking about before as with adding a leaving group, so that you can render the carbohydrate to become a electrophile, and then it just adds to it the same way. So lastly, this last flow chart that I've got here is to show really 3 main types of lipids: simple lipids, complex lipids, and derived lipids, and to talk about sort of different examples of each. So let me tell you about the basic definitions of all 3 of them, and then we'll talk about more about the specifics that we can go from there. Simple lipids are really just esters of fatty acids with various alcohols. Alcohols meaning any kind of R group, it can be anything you want it to be. And so really there's only really just 2 types. You can either have triglycerides or wax. Triglycerides, again, we've already talked about it. Again, and based off of whether it's long or it's short, or if it's saturated or unsaturated, determines whether it'll be solid at room temperature, which you would call it solid fat, or it's liquid at room temperature, which you would call a liquid fat, or just an oil is another way of putting that. A wax, on the other hand, is an ester formed from a long chain fatty acid plus a long chain alcohol. And because of this, it has a higher melting point than triglycerides, which is why you really can't get it to be liquid, really, is what that means. And so essentially what that means, what I just said, is essentially you just have a long chain, meaning alcohol, meaning you have the alcohol at the end of a long hydrocarbon chain, reacted or added to a long chain fatty acid. So just like a normal fatty acid, really. Secondly, we have complex lipids. Complex lipids are, or also sometimes compound lipids, are really esters of fatty acids with alcohols. In other words, just like we said before. But it also contains additional groups, such as phosphate groups, or nitrogens, or carbohydrates, or proteins, or sphingosine, any of these other things. And so we've actually already talked about these two right here, the phospholipid and the glycolipids, which are both derivatives of complex lipids. The phospholipid has to do with the phosphate group, and the glycerophospholipid has to do whether if this phosphate group and the fatty acid are using a glycerol backbone, which we already talked about, and the sphingophospholipid has to do with whether it is using a sphingosine backbone. So that's really the difference between those two. Both of which, again, having a phosphate group is just different, and fatty acids is just a different backbone. And then glycolipids, like I said before, instead of having a phosphate group added to your lipid, you can actually add a monosaccharide or polysaccharide, and like I said before, based off of whether it's one or many. If it's one, it's a seroside, and if it's many, it's gangoside. And again, the sort of idea you can remember that by is that multiple is gang, and it's just in the name, I guess. So this other part right here, I think I mentioned at this very beginning of the video, a lipoprotein. A lipoprotein is essentially a complex structure of a protein and lipid, such that it is capable of carrying non-water soluble molecules such as lipids through the bloodstream to its target tissues. Because again, what is the bloodstream made mostly of? Water. And then what are lipids? Not soluble in water. So therefore you have a bit of a problem. And the idea of the lipoprotein is that it kind of just solves just that. It carries the lipids through the bloodstream to wherever it's supposed to go. And so when we think about the different types of lipoproteins, we really have just three types, kind of. We have HDL, VLDL, and chylomicron. HDL stands for high density lipoprotein. VLDL stands for very low density lipoprotein. IDL, which is going to be something that you can produce from VLDL, stands for intermediate density lipoprotein. It's the in-between between very low and low. And then lastly, from intermediate density lipoprotein, which is a precursor, you can produce very low density lipoprotein. So it's kind of like a chain of events. Like you can produce this from this to this. So chylomicrons, the idea of it is that it transports dietary lipids from the intestine to peripheral tissue, and it does this through the lymphatic system. The idea is that the dietary lipid is just too big, too bulky, that sort of thing, to go directly into the capillaries from the intestinal wall lining. So essentially we have this special sort of pathway that it can follow, specifically with this chylomicron, that's what that is, such that you can carry the dietary lipids such as triglycerides or cholesterol, which again are too big, through the lymphatic system first, which empty into the junction between the subclavian vein and the jugular vein, so that you can enter into the bloodstream from there, from that point. That's its idea there. HDL, which stands for, again, or let me back up. So LDL and HDL, they're kind of two opposites of each other in a way. HDL, or in other words, high density lipoprotein, is thought of in terms of as being good cholesterol, and then low density lipoprotein is thought of as being bad cholesterol. They are kind of two opposites of each other in the sense that LDL, or low density lipoprotein, transport cholesterol, and again, they both transport cholesterol. They're kind of just transporting from one place to the other, kind of like opposites of each other. LDL transports cholesterol from the liver to tissue. HDL transports cholesterol from the tissue or peripheral tissue to liver. So they're kind of opposites, like backwards of each other. And then VLDL, or also called very low density lipoprotein transports specifically endogenously synthesized lipids from the liver to tissue. And then intermediate density lipoprotein is just like the intermediate, so between. And then lastly, the third final category of lipids is derived lipids. Derived lipids is essentially just any kind of derivative obtained by the hydrolysis of the previously stated ones, simple and compound lipids. So in other words, what that means is that you can take any of these aforementioned lipids and you can just hydrolyze them. In other words, break them down and get what they were made of, and that's just what derived lipids are. These are just what you get whenever you break it down. And so really we have two broad categories we're going to talk about. We're going to talk about derived lipids in terms of either being arachidonic acid and its derivatives, or we're going to talk about cholesterol and its derivatives. So first, what is arachidonic acid? And by the way, this is what that is right there. Arachidonic acid is it's made of 20 carbons, and it is a polyunsaturated fatty acid with 4 cis double bonds. I'm fairly certain that earlier on the very first sheet, we were talking about the delta system for designating the number of carbons and the number of unsaturation and where those unsaturated double bonds occur, like the naming system we're talking about at first. I'm pretty fairly certain that we did mention arachidonic acid as being an example of one of those. But essentially what arachidonic acid is a derived lipid which serves as a precursor to a broader category of signaling molecules called eicosanoids. And eicosanoids I don't think they're technically considered hormones in the sense that the classical hormone is traditionally thought of as being the signaling molecule that travels the bloodstream and circulates throughout systemic circulation until it reaches a tissue from far away. Eicosanoids are kind of just like signaling molecules that are really more so autocrine and pericrine, meaning they're kind of like hormones. They produce similar effects to hormones, except for the fact that their effects they're not very long lasting, so they don't last that long, in the sense that they have a short half-life, and in the sense that autocrine being that the cell can produce the eicosanoids and it can act on itself, like it can basically talk to itself, or it can function as a pericrine signaling molecule in the sense that it only sort of communicates with surrounding tissue as opposed to tissue that's really far away, like you would think with traditional hormones. In general, eicosanoids have a lot to do with really just immunity and inflammation and that sort of thing, whether it be for inflammation or against it, like anti-inflammation. And so when we think about, there's really two of these main categories that derive from arachidonic acid, which are leukotrienes and prostaglandins. Now, their name kind of does indicate a little bit about its history. Leukotrienes, the leuko kind of indicates kind of like its function in the sense that has mostly to do with leukocytes, which are white blood cells. And then the triene part has to do with the fact that its chemical structure is a triene, meaning it's conjugated and has 3 double bonds. And a leukotriene has mostly to do with leukocytes in the sense that it has a lot to do with, it's produced for allergy attacks and all of the other side effects associated with it, such as like inflammation, airway constriction, mucus production, edema, that sorts of things. And then secondly, we have prostaglandins. The reason why it's called the first like prefix sort of is prosta is because it was originally isolated from seminal fluid, and it was just originally thought of being as only produced from the prostate gland. However, that's later found out that is not like the case. It's not that exclusive. But from that, you can get 2 more categories: thromboxans and prostacyclin. Thromboxans kind of just hence the name. The thrombo part has to do with blood coagulation and blood clotting. Thromboxans have to do, and these two are actually kind of opposite to each other. So thromboxans are produced by platelets whenever there is a need for it, and of course they cause vasoconstriction, platelet aggregation, and all the other things associated with blood coagulation. And the prostacyclins are these kind of like the opposite. They, like I said before, thromboxane induces vasoconstriction and platelet aggregation, Prostacyclin kind of does the opposite. It does vasodilation, inhibits platelet aggregation, so in other words, the platelets like sticking together. And it also inhibits the release of other inflammatory mediators. The idea is that the endothelial cells of blood vessels are constantly producing prostacyclin, and whenever the endothelium is damaged, the balance sort of shifts between prostacyclin and thromboxane. Okay, so lastly, I want to talk to you about cholesterol and its derivatives, which are steroids, hormones, which based off of wherever it's produced, whether that's in the testes, the ovaries, or the adrenal cortex, or any of those other things, will determine kind of what kind of steroid hormone that you produced from the cholesterol precursor. Also, another thing about cholesterol I just wanted to point out, I guess, is that cholesterol is a very sort of intricate part of cellular membranes, and the more cholesterol you have, the more rigid it is, which has to do with a lot with the fluid mosaic model, which is just a model or like a way of understanding cellular membranes as being these fluid, as being like on a scale of fluidity, and this scale of fluidity has a lot to do with temperature. For example, kind of like with salmon have a special thing where they can actually influence the fluidity of their membranes by either adding or removing polyunsaturated fats from their membrane to cause it to become more rigid or less rigid according to the environmental heat. The idea is that if it's really cold, the salmon will make the membrane more fluid, because by being cold, this lack of heat, this coldness, will cause it to become more rigid and unstable and not work. And it does the opposite whenever it's hot. It will make it more rigid by adding or removing the polyunsaturated fats from the cellular membrane. So let's talk about cholesterol. So cholesterol, before I talk to you about all these different sort of like intermediates with cholesterol, I want to introduce you to the fact that central metabolism, like we said before, is basically this process by which we take glucose and we convert it into a more usable energy source, adenosine triphosphate, also called ATP. And throughout this process, we have a lot of intermediates as it undergoes further oxidation and phosphorylation. And the idea is that somewhere along the sort of process of central metabolism, we can take certain intermediates and certain numbers of these intermediates and sort of add them to each other in these chemical processes to biosynthesize cholesterol. And the idea is basically that we have to take, it's like we have like these little building blocks, and each building block, you can think about it in terms of having X number of carbons, and then we add them to each other such that we can finally get our final product. And the idea is that we're just trying to make it bigger and bigger in a specific way, and the final product is able to kind of like has like free rotation on some of their bonds, so that it can distort its shape and kind of react with itself in a way to form the rings necessary for cholesterol. So first we have acetocoenzyme A, or also called acetyl-CoA as we've got it written here. The idea is that acetyl-coenzyme A is one of the intermediates in central metabolism. You get it from pyruvate, and then from which you will react acetyl-coenzyme A with oxaloacetate to produce citrate. And then, like normal, you would go along it and do your thing with oxidation and so on and so forth. That's not the point. The point is that acetyl-coenzyme A is one of the intermediates you get from sugar, and essentially to create acetyl-coenzyme A, which is the next part. Again, like I said, I've got in blue written all the number of carbons. That's what all these mean. So 2, 4, 6, 6, 5, 10, 15, 30, 27. Those are all the number of carbons in each one of these steps along the way. Essentially, all you got to do is to create acetyl-coenzyme A is you react two acetyl-coenzyme A's together. So in other words, 2 + 2 is 4. So you just take 2 of those and you can create acetyl-coenzyme A with that. And then to create HMG coenzyme A, you kind of do something similar in the sense that you take what you just made, the acetyl-coenzyme A, and you add another acetyl-coenzyme A, so that you get that 4 + 2 = 6. And then to get mevalonic acid from HMG coenzyme A, essentially all you're going to do is you reduce it with an enzyme And one of the interesting things about statin drugs, which statin drugs are, their purpose is to reduce cholesterol, is that this is actually the target of statin drugs, in the sense that they're trying to inhibit the enzyme from reducing HMG coenzyme A to mevalonic acid, or mevalonate. And so again, since it's just being reduced the same number of carbons, but when you get to isopentenyl pyrophosphate, or IPPP, which has one less carbon, what actually happens between there is you have phosphorylation and decarboxylation. And then from there, to get GPP, or geranyl pyrophosphate, you just add 2 IPPs, or which is what we had previously, to each other. So like 5 plus 5 equals 10. And then to get to the next step, you're going to add what you just had, the GPP plus the IPP one more time. So 10 plus 5, and you get 15. And then to get squalene, you just kind of do the same thing, in the sense that you just created the FEP, or the farnesyl pyrophosphate, and then you add them to each other, like 2 of them added to each other, to get squalene. And from squalene, you actually have one more intermediate. I didn't write it down though, because I was running out of room. But essentially from squalene, you go from that to cholesterol via the fact that squalene has, again, like I said, it has rotation, it has free rotation on some of the bonds. So basically the shape gets distorted such that it reacts with itself to form the rings necessary for cholesterol. And cholesterol, as well as all of the steroid hormones, follow the same basic pattern I have written down right here, which is the ABCD model. Basically, you have these there's 4 rings in total, like you could see, all of which are cyclic. The first 3 ABC are all 6-membered rings, and then D is 5-membered rings. And different steroids what makes different steroids different from each other, or unique, or have their own unique chemical properties, is based off of whether they all have the same basic structure, but there are different substituents added at different places with different stereochemistry, as well as the rings themselves could be unsaturated, so they have double bonds. They could be aromatic, or something else like that. They could be conjugated, something like that. That's how you get the sort of special properties of it. And then from cholesterol, once you have cholesterol by biosynthesizing the different intermediates from central metabolism, from there you can get a number of things. Vitamin D, you can get bile salts. Bile salts, of course, are just what's produced from the liver, stored in the gallbladder, and then from the gallbladder through a duct to the stomach to emulsify the whatever food you ate. Again, bile salts are amphipathic, meaning they have a hydrophobic and hydrophilic end. And essentially the idea is that they form a miscule, I think that's how you pronounce it, around fats. Again, their target molecule is for fats, and they just form this structure around fats such that their hydrophobic ends are oriented towards the fat, and their hydrophilic ends are oriented outwards. Because again, the reason why the hydrophobic ends are oriented towards the fats is because the fats themselves are also hydrophobic, so you have to have like dissolve with like, so that they're facing each other. And then from bile salts, you have primary. I haven't written it down specifically. We have primary and secondary bile salts. Essentially, what differentiates the 2 of them is that secondary bile salts can only be produced by some of the gut bacteria, whereas primary are produced by you yourself, your liver. And that's just one of the reasons why you need gut bacteria.